Hi, my name is Louis Back, and my project concerns the developmental changes in C. elegans in which the E2 ubiquitin-conjugating enzyme coding genes have been suppressed. Ubiquitin is the protein that determines which proteins are destroyed or kept. It determines this through a system called the ubiquitin proteasome system. It consists of three stages called the E1, E2, and E3 stage. In this case, I focused on the E2 stage in a small nematode called C. elegans. To suppress the E2 ubiquitin-conjugating enzyme coding genes, I used a process called RNA interference, or RNAi. RNAi uses double-stranded RNA, which is then introduced into an organism where it is used to create a protein that destroys the target mRNA, suppressing the gene. The genes suppressed in this case were UNC22, UBC6, and UBC7. The negative control introduced a gene that coded for a fluorescent protein called GFP, which should not change any other physical attributes of the worms. The UNC22 gene, if suppressed correctly, should cause worms to twitch. The UBC6 and UBC7 genes are both genes that code for the E2 ubiquitin-conjugating enzymes. My hypothesis was that worms would not be able to develop correctly, which would lead to less worms or deformed worms. Suppressing the UNC22 gene in one group made 67.9% of worms twitch and 69.6% in another group. Meanwhile, the GFP wild type had no twitching worms for either group. Here is an example of wild type worms. And here is an example of worms whose UNC22 genes have been suppressed. Here is another example of a worm whose UNC22 gene has been suppressed. As you can see, its head is twitching as it struggles to move. Here, the average speeds in millimeters per second for each group is graphed. The UBC7 worms are fastest on average, while the UNC22 worms are considerably slower. When subjected to a t-test, however, the results indicate that the sample size is too small for a statistically significant finding. Here is a sample of UBC7 worms. As you can see, they are visibly more active than the GFP worms. And here is a sample of UBC6 worms. Their speed seems almost the same as the GFP worms. In response to the hypothesis, there were no notable changes in the development of the worms other than in speed, and a larger sample size is required for the statistically significant data for the UBC6 and UBC7 experimental groups.